my name is Jennifer Benson, and I will be talking about how our bodies get the energy from the foods that we eat. Throughout the day, we have food intake into our stomachs, such as apples, so fruits and vegetables, or even a hot dog like protein. And how does our body take that food and turn it into energy that we can use for or on a daily basis? The main reason that we eat food is for energy in our bodies. Food is our body's only energy supply, but the only way that our bodies can take the food that we eat and convert it into usable energy for the body is through a process called cellular respiration. Breaking down the cupcake in the body would be an example of food catabolism. The degradation of glucose, the citric acid cycle, and the electron transport chain are important components to ultimately create ATP, which is an energy source to couple and drive unfavorable reactions. So just a summary of cellular respiration, there are 3 steps as I just stated. The first one is glycolysis, which is the breakdown of glucose. The second step is the citric acid cycle. The third step is the electron transport chain, and in the next slides you'll see a summary of glycolysis, the first step of cellular respiration. The word glycolysis means glucose splitting, which is exactly what happens in glycolysis. Enzymes split one molecule of glucose into 2 molecules of pyruvic acid, and this is a series of 9 steps. So the first step, the glucose ring, is phosphorylated, which is the process of adding a phosphate group to a molecule that's taken from ATP. So at this point in glycolysis, 1 ATP is consumed. This first reaction uses the help of hexokinase and atomic magnesium. This first step results in glucose 6-phosphate. So glucose is converted into glucose 6-phosphate at the end of this step. Step 2 of glycolysis is the conversion of glucose 6-phosphate to fructose 6-phosphate. This reaction uses the enzyme phosphoglucose isomerase. The reaction involves the rearrangement of the carbon-oxygen bond to change the 6-membered ring to a 5-membered ring. So you can see that in the diagram below. Moving on to the third step. The third step is the conversion of fructose 6-phosphate from the previous step to fructose 1,6-biphosphate. Similar to step 1 of glycolysis, ATP is used to provide the additional phosphate group. And then the enzyme used, as you can see in the diagram below here, is phosphofructose kinase. In the fourth step of glycolysis, the enzyme aldolase catalyzes the cleavage of FBP and yields 2 3-carbon molecules. And the 3-carbon molecules are GAP and DHAP. So the only molecule that continues on in glycolysis is GAP. However, DHAP is acted on by another enzyme, and then it's converted to GAP so it can continue on in the process. So we just reviewed step 1 through 4. That is the first half of glycolysis. So we now have 3 carbon molecules, but we've not yet fully converted 1 glucose molecule into pyruvate. So moving on to step 5, the second half of glycolysis, 2 main things happen. One of them is that GAP is oxidized by coenzyme NAD, and the second thing is that the molecule is phosphorylated by a free phosphate group. And this reaction includes the enzyme of GAPDH, as you can see in the small diagram below. Step 5 ended with 1,3-biphosphoglycerate, and in step 6, this molecule is converted into 3-phosphoglycerate by an enzyme called PGK. Unlike the other reactions, this one loses a phosphate group, and the phosphate is transferred to ADP, and that yields our first ATP molecule. Another ATP is also created in this step. So we used 2 ATP in the previous steps, and now we just gained 2 more. So now we're at a net gain of 0, or a net of 0 ATP. In step 7 of glycolysis, 3-phosphoglycerate becomes 2-phosphoglycerate by rearranging the position of the phosphate group. The molecule that catalyzes this reaction is called PGM. The 8th step converts 2-phosphoglycerate into PEP. This reaction is catalyzed by the enzyme enolase. Enolase actually removes a water group and dehydrates the 2-phosphoglycerate to form the PEP. So as you can see in the diagram below, we get the PEP plus the water that was dehydrated from it. The 9th and final step of glycolysis is this, and here using pyruvate kinase enzyme, PEP is converted into pyruvate. As the name of that enzyme suggests, this reaction involves the transfer of a phosphate group, and this step itself yields 2 ATP. To recap the first stage of cellular respiration, called glycolysis, we have turned one molecule, as you see at the top of this diagram, one molecule of glucose all through the 9 steps, and then at the bottom we have gotten 2 molecules of pyruvate. We generate 4 ATP throughout this process, and we use 2 ATP throughout the process. Therefore, we have a net gain of 2 ATP from glycolysis alone. The next step of cellular respiration is the citric acid cycle. In the citric acid cycle, the pyruvate molecules that were made in glucose, as you see in the cytoplasm below, are transported from the cell's cytoplasm to the mitochondria. Now, obviously, the size of this pyruvate molecule is not to scale, but otherwise you wouldn't have been able to see it. So, the citric acid cycle does consist of 8 steps, and these are complex chemical reactions. As you can see in the diagram below here, the pyruvate molecule from glycolysis loses carbon dioxide to form acetyl coenzyme A, which is a 2-carbon molecule. Acetyl coenzyme A is the molecule that enters the citric acid cycle. During the citric acid cycle, acetyl coenzyme A is attached to oxaloacetic acid to form citric acid. This cycle's 8 chemical reactions require the participation of enzymes and other molecules. Here's another, and I believe pretty simplified diagram of what's going on in this step. So the citric acid cycle is going to continue around and around and around and keep going on until as long as NADH and FAD are present. So for each pyruvic acid molecule that enters the cycle from glycolysis, 4 molecules of NaH are energized to form 4 molecules of NADH and hydrogen, and 1 molecule of FAD is energized to form 1 molecule of FADH2. These energized molecules are used to power the 3rd phase of cellular respiration, the electron transport chain. The electron transport chain is the final and most important step for generating ATP in cellular respiration. This is the stage that forms the most ATP, and as well as the citric acid cycle, it occurs in the mitochondria of the cell. The electron transport chain creates a proton gradient by moving electrons from high energy to low energy. This proton gradient powers ATP production and not the flow of electrons. Looking at the diagram here, you can see that electrons are passing from complex to complex and you can see that by following the blue arrows. And it's powering the movement of hydrogen atoms, and you can see that represented by the pink arrows. And they're going into the inner membrane space. Using the NADH and FADH2 created from the citric acid cycle, electrons are donated to the transport chain. This is how a net gain of 32 ATP is reached from this stage of cellular respiration alone. In addition to all the ATP or energy that's produced, it also produces H2O, so water, NAD, and FAD, and those two molecules, NAD and FAD, will be recycled and used again in the Krebs cycle. So here you see a chart, just reviewing the stages of cellular respiration. We have glycolysis, citric acid cycle, and electron transport chain. And then in the other column here, you can see which stages are responsible for producing how much energy. So glycolysis is responsible for 2 ATP, citric acid cycle is another 2, and then the electron transport chain is 32 to 34. So you can see how the energy from the cupcake that you eat is converted, and you get all this ATP just by going through this the cellular respiration. So, to summarize and review the main question that we're answering throughout this presentation, as we eat food such as the cupcake that was used in the example, and it goes into our body, what happens so that we can use the food to create energy for all of our body? That is cellular respiration. So just to summarize the process of cellular respiration one more time, we start off with one glucose molecule that goes into glycolysis. From glycolysis, we're getting 2 ATP and 2 molecules of pyruvic acid. That pyruvic acid molecule is entering the Krebs cycle or the citric acid cycle, and from that we're getting 2 ATP. And then we go on to the electron transport chain, the third and final step of cellular respiration. And the electron transport chain shuttles electrons down a series of redox reactions that release energy and create 32 to 34 ATP. And that is how our body generates energy from our food that we consume.